Hey, it's Ned. Welcome to Tag Tutors, where I'll be showing you step-by-step guide on how to ace GCSE equation. This applies for all specifications: AQA, EdXL, OCR, doesn't matter. Get a pen, a paper, and listen. A student placed 3 different sized cubes of agar jelly into separate beakers. Different sized cube of agar jelly into different beakers containing the same concentration of hydrochloric acid. The cubes contained a pink indicator. The indicator becomes clear when in contact with acid. Figure 2 shows the result of the investigation after the cubes had been in acid for 120 seconds. Cube A, B, C. Question: the distance from the outside of cube B to the pink area was 3 millimeters. Calculate the distance diffused by the hydrochloric acid in 1 second. So 1 second, 3 millimeters. The cubes had been in acid for 120 seconds. So 120 seconds. My first step, how many marks? So this is 2 mark. So I need to show working out. Okay, for 1 mark. Then my answer would be 1 mark. Okay. So my working out would be, if in 120 seconds the distance was 3 meters, we're looking for 1 second. So what do I do? 120 seconds is 3 millimeters. So I'm looking for 1 second. 1 second, that would definitely be less than 3 millimeters. So applying my maths skills, it would be 3 divided by 120. That would give me 0.025 millimeters. So my answer would be 0.025 millimeters. Always remember to write down your answer on the line. When it says calculate, means you need to use the values that you have to write down your answer. Okay, don't leave your answer here. Your final answer should be on this line. A plant leaf cell is 0.04 millimeter long. 0.04 millimeter long. Calculate the length of the image after the cell has been magnified 500 times. Magnified 500 times. Step 1. How many marks is the question? Question is what? 2 marks. Step 2. The command word here says calculate, so I need to use values to find answer. Okay, my first step would be show my working out. Show work. My second mark would be my answer. So what do I know about magnification? I can remember my triangle, so I draw that by the side. Okay, this is what I would do in an exam. I know that I am, so image size, actual size, magnification. Okay, so I am looking for the image size. So the image size would be actual size times magnification. So I would be A times M, and actual size here is 0.04. So 0.04 millimeters times magnification is 500 times 500. My answer, this step gets me 1 mark. My answer would be 20 millimeter. This gets me my final mark. A scientist obtained a mass of 0.0062 nanograms of DNA, 0.0062 nanograms of DNA from a diploid human cell. Remember, diploid means 2 pairs. Calculate the mass of DNA the scientist should obtain from a haploid human cell. Haploid human cell has 1 set. Now, my first step, question is 2 mark. So that means I need to 1 mark for show work, 1 mark for answer. Okay, my step 2, the command word in this is calculate. Calculate means use value to find answer. 1 nanogram is equal to 1000 picograms. This is very important because my answer should be in picograms. Okay, we'll come back to that. Diploid cells are 2 sets. Haploid cells are 1 set. Remember, haploid cells are found in gametes, and that's just 1 set. Okay, so what do? We need to divide this value by 2. That gives us a haploid cell. So 0.0062 nanograms divided by 2, that gives us 0.0031 nanograms. Okay, so that's our first step. Now, 1 nanogram is equal to 1000 picogram. Okay, so I put this value here. What if I'm looking for 0.031 nanograms? 1 is much bigger than this, so my answer should also be a lot smaller than 1000. So what do? 0.0031 times 1000. That gives me an answer of 3.1 picograms. So here I have to write down my answer here, 3.1 picograms. Okay, so 1 mark for this, 1 mark for my answer. Devise a method using cubes of agar jelly to investigate how temperature affects the rate of diffusion. The keyword here is devise. Devise a method using cubes of agar jelly to investigate how temperature affects the rate of diffusion. Step 1. How many marks is the question? This question is what? 3 marks. So I need to give 3 different points. That will get me 1 mark each. My step 2, the command word here is devise. Devise means I need to use my knowledge to show how I would carry out an experiment. That means set out how something will be done. So my first step would be, I'm looking for temperature. So if I'm going to use temperature, so I need to work at different temperatures to find out if the rate is slower in maybe a reduced temperature or if it's faster in a high temperature. So I need different temperatures. So my first step would be heat acid, because I need to use an acid, to different temperatures. Or if I don't want to say that, I can use heat the agar jelly cubes to different temperatures. So that will be the same point. Okay, when that is done, my point 2 would be use the same size agar jelly cubes. So I am keeping this the same. This will be my control variable. Likewise, I need to use the same volume, or this is another the same point, so I can say this or that, the same volume or concentration of acid. My last point would be carry out the experiment for the same amount of time. Lastly, I need to measure something. Okay, I have changed the temperature, I need to measure something. So my last point would be measure clear distance at each temperature. Okay, so we are changing the different temperatures, so this would be our independent variable. What we measure will be our dependent variable, and we are keeping these two the same size agar jelly, the same volume of acid, the same carry out for the same amount of time. So I have my 3 variables in this experiment. Figure 4 shows some carrot sticks. You can see some different sized carrot sticks. A student chose 3 carrot sticks and weighed each one. 3 carrot sticks, weighed each one. The carrot sticks were placed in 50 centimeter cube of distilled water. After 2 hours, the student weighed each carrot stick again. Figure 5 shows the results for these carrot sticks, P, Q, and R. Carrot stick P, Q, and R. Mass at the start, mass after 2 hours, as change in mass. Give one reason why the student used 3 carrot sticks instead of 1 carrot stick. Step 1, how many marks? So this is 1 mark question. That means I need to just give one answer. Why the student chose to use 3 carrot sticks instead of just 1. I can say that a single result could be anomalous. Or I can say the results could be the same, and because if I use more than 1, I can calculate a mean. Using more than 1 carrot stick, the student can calculate the mean value. Give 2 ways that this method could be improved. So instead of the student just choosing random 3 carrot sticks, the student could cut sticks from the same carrot. Secondly, the student could dry the carrot sticks before weighing them, because if you don't dry the carrot sticks before weighing it, that result of mass will be wrong, as there will be water inside the carrot sticks before weighing. So 1 mark for this, 1 mark for that, and that will give me my complete 2 marks. Calculate the percentage change in mass of carrot stick Q. Use the equation percentage change in mass divided by mass at the start times 100. Give your answer to 2 significant figures. Step 1, the question is what? 3 marks. So my first mark would be show work. My second mark would be for my answer. My third mark would be to put my answer, to 2 sig figs. And step 2 says calculate. So I need to use the values given to me. If I bring back this table, we are looking at carrot stick Q. So we're looking at this value. So we can see the mass at the start is 4.2. The mass after 2 hours is 5. The change in mass is 0.8. Change in mass is 0.8. Mass at start is 4.2 grams. So I put these values into this equation. That will give me 0.8 divided by 4.2 times 100. My answer would be 19.048. This gets me 1 mark. My working out gets me 1 mark. Okay, my third mark, I need to put my answer in 2 sig figs. Okay, 2 sig figs will be 1, 2. Figures, figure 1, figure 2. So my answer would be 19%. Explain the change in mass of the carrot sticks. My step 1, how many marks is the question? This is a 2 mark question, so I need to give a point and give a reason for that point. That gets me 1 mark each. Step 2, the key word here is explain, which means I need to give a reason. So my first point on why there was a change in the mass of the carrot sticks. The carrot stick gained mass because, my second point, water moved into the carrot cells through osmosis. The development of electron microscopes has increased. Our understanding of cells and their features. Figure 8 shows 2 images of ciliated epithelium, one taken using a light microscope, one using an electron microscope. Explain how the electron microscope image helps us to understand more about the ciliated epithelium, electron microscope. Step 1, question is 3 marks. So I need to give a point, give a reason, or give 2 reasons, or give 2 points, give 1 reason, however you want to do it. Step 2, explain means give a reason. Why would I use an electron microscope? That is because it has a higher magnification. So, electron microscope has a higher magnification. Okay, now what is the reason behind this? So that the cilia are more visible. Another reason could be number 3, and the subcellular structures are more visible. Okay, more visible. I have a point that gets me 1 mark, and I have 2 reasons for that point. That gets me my complete 3 marks. Many cells contain an enzyme called catalase. Okay, underline it. Catalase breaks down hydrogen peroxide into water and oxygen. Water and oxygen. A scientist investigated the effect of hydrogen peroxide concentration on the time taken to produce 20 centimeters cube of oxygen. Figure 6 shows the equipment used. So from the figure, I can see my flux, I can see my catalase, my 25 centimeters cube of hydrogen peroxide, I can see a test tube, I can see bubbles, 20 centimeters cube of oxygen collected. The question says, state how the scientist could control the temperature of the flux. The scientist could use a water bath. Okay, so that will be my answer. Scientist could use a water bath because that is what I have used in schools when I'm carrying out an experiment and I want to control the temperature. The easiest one to use is a water bath. That gets me 1 mark, and that also means that I've stated how. Number 2, explain why the temperature should be controlled. Why should we control the temperature in this investigation? Step 1, question is what, 3 marks, so I need to give 3 points. Step 2, the command word here is explain, which means I need to give a reason. Give a reason, so make 1 point or give 2 reasons. It says here clearly, many cells contain an enzyme. What do I know about enzymes and temperature? I know that enzymes have an optimum temperature. So that could be a reason why the scientist would want to control this. So my point would be, enzymes have an optimum temperature. If the temperature is too high, the enzyme active site will do what? It would denature. Because, point 2, remember, if you're writing an explain answer, you have to use the word because you're giving a reason. If the temperature is too high, or I can say increases up to the optimum temperature of the enzyme, the active site of the enzyme will denature. Okay, so that's my first reason. Second reason is because temperature affects the rate of enzyme activity. Okay, so that is why we need to control this temperature. Now, some people in an exam will leave these 2 points and think this will get you 2 marks. No, we wouldn't. This is one reason. The enzyme's active sites will denature. That's one reason. Second reason is because the temperature affects the rate of enzyme activity. Okay, so I have 1 mark, 1 mark, 1 mark. This investigation used 5 different concentrations of hydrogen peroxide. You can see 4, 8, 12, 16, 20. And the time taken to collect 20 centimeters cube, 32 seconds, 14 seconds, 9, 7, 6. So the time is decreasing as the concentration is increasing. Okay? Concentration increases, the time will decrease. Alright, so that's what I can tell from table. Cool. Let's get to the question. The question says, complete the graph by plotting the points and drawing a line to show the trend in the data. Complete the graph by plotting the points. Draw a line of best fit to show the trend. This question is 2 marks. And when it says complete the graph, that's a straightforward command. Draw on this graph. At concentration of 4, the time taken was 32. That has been done for me. You can see that has been done for me. Okay, so I'm looking at my next value. Next value will be concentration of 8. So 8 is over here. And at 8, the time taken is 14. 14, so where is 14 on this scale? It is always difficult for students to read scales. So this is 10. So this will be 11, 12. So this will be 11. So will be my 12, 13. So this will be my 14. And at 12, my value is 9. So 9 would be here. At 16, my value is 7. So I have 5, 6, 7. At 20, my is 6. So this is 5, this is 6. Now, someone will ask, why am I skipping 2 boxes for my 1? This is because I have 10 boxes and this is moving up in 5s. 10 divided by 5, that gives me 1 value for 2 boxes. Okay, 10 boxes divided by, the scale is 5, difference here is 5. So I do 10 over 5, that gives me 2 boxes is 1. So 1, 2 boxes gives me 6. 1, 2, then 7. 1, 2, then 8. 1, 2, then 9. That's how to read the scale. Okay, so now I have to plot my line of best fit. I can see that is a smooth curve. So I have to join that up with my 32. So I have to draw a smooth curve. So that would be my 2 mark answer. Describe the trend shown in the graph. Use data from the table in figure 7 to support your answer. Step 1, this question is 3 marks. 3 marks, I need to give 3 points for 1 mark each. And it says describe, so I need to state facts. Remember when we went to the table, I found out that as the concentration is increasing, the time taken is decreasing. And I'm using the actual words used on the table. Concentration of the hydrogen peroxide, time taken to collect 20 centimeters cube of oxygen. As the hydrogen peroxide concentration increases, the time taken, so I'm not just writing time, the time taken to collect, use the actual words, to collect 20 centimeters cube of oxygen decreases. My second fact will be, so looking at this graph, I'll bring back the graph. Looking at this graph, I can see that the curve would flatten out over here. Okay, so that's the fact that I can see, so I have to write it down. The curve flattens out. My point 3, where does the curve flatten out? So the curve flattens out after 20. So I would say the curve flattens out after a concentration of 20. So I have stated the facts from the table and the graph, because it says use data from the table and the graph. Okay, a student cut a piece of onion and placed it on the microscope slide. A piece of onion, on a microscope slide. The student then placed this slide on a stage of a light microscope, and looked through the eyepiece. No cells could be seen in the piece of onion. No cells could be seen. Explain 2 ways this method could be improved. 2 ways method could be improved to see the details of the onion cell. This is a 4-mark question. That means I need a point, a reason, a point, a reason. That carries 1 mark each. Explain is the command word. Explain means give reason for my point. So my first point would be, it says a student cut a piece of onion. That's not how to do it. The student needs to cut a single or a thin layer. Okay? Students should cut a single or thin layer of cell. Why do we need to cut a very thin layer? That is because so that light can't pass through the cells. Remember, we are using a light microscope. Using a thin layer, light can easily pass through. Okay, light can pass through the cells easily. Secondly, the students did not add any stain. We need to use a stain. Use a stain. You can name the iodine or whatever you want to say. So use a stain. I keep it simple. Why do we use a stain? Because staining the cell ensures that the structures inside can be seen properly. That is why we use stains during experiments. So I've given a point, a reason, a point, a reason. 1 mark, 1 mark, 1 mark, 1 mark. I could also add many other points in this. I could say adjust the focus on the microscope. Why is that? To see the cells clearly. I could say that as well as a point and a reason. Or I could say select a higher power lens. Why am I doing that? To increase magnification. So there are many points that I could say in this part of the question. Figure 1 shows 3 cells. You can see a bacterial cell, you can see a plant cell, and animal cell. Okay, bacterial cells and plant cells, they have the same structure X, but animal cell doesn't. What is structure X? Structure X would be a cell wall, because animal cells don't have cell wall. That gets me my 1 mark. The bacterial cell in figure 1 has a flagellum. State the function of the flagellum for 1 mark, and to state and to give a reason or give a function. So my function will be to swim or move. Give 1 other difference between the bacterial cell and the animal cell. Bacterial cell have no nucleus. Or I can say bacterial cells have a chromosomal DNA. Figure 6 shows a diagram of a cell. Which row of the table identifies both structure P and structure Q? P would be the vacuole because it's big. It's always big on the plant cell. And Q would be the cell membrane because it's the inside structure. For 1 mark, my options are A, nucleus and cell membrane. No. Nucleus and cell wall. Okay. No. Vacuole and cell membrane. The answer would be vacuole and cell membrane. So that would be my answer. Plant cells have a cell wall and a large vacuole. Draw 1 straight line from each structure to its function. 2 points. So 1 line is for 1 mark. The other line is for 1 mark. Cell wall. What does a cell wall do? It contains, where respiration occurs. No, it contains cellulose. Cellulose cell wall. Okay, yes. Cell wall contains cellulose to provide support as well. That's correct. Large vacuole controls the cell. No. Where? No. Stores cells up. Yes. So this is the answer. If I don't know the answer, I'm using the process of elimination to find the correct answer. The tips of plant roots are where many cells are dividing by mitosis. Which term describes the area of a root where many cells are dividing by mitosis? Step 1 is a 1 mark question. So I need to choose the correct option. Option A, meristem. Option B, root hair cell. Option C, xylem. Option D, phloem. No, that would be a meristem because I know meristems divide to form many other cells. Okay, just like the stem cell in humans. Plant root cells contain an enzyme that joins glucose molecules together to make starch. Devise a plan to investigate the effect of pH. The effect of pH on the activity of enzyme. Step 1, this is a 3-mark question. So I need to give 3 different steps for my experiment. The command word here is devise. So say how something works. So what step would I use for my experiment? My first step, I am using enzyme. Okay, I'm using an enzyme. So what would I do? I would add enzyme solution to glucose because it says enzymes join glucose molecules together to make starch. So I'm adding enzyme solution to glucose to see if it would make starch. Okay, right, step 2. I need to test that starch has actually been made. So that would confirm that this statement is correct. So I need to test that starch is present when I have added this together. 1 mark, 1 mark. Okay, so I have tested that starch is present. Now I need to time it. Time how long it took for starch to become present. Why am I doing this? To become present? I'm doing this because I am going to change the pH of the enzyme solution. So my last step would be use different pH values for my enzyme solution and repeat the time. Okay, so using different pH values and find out how long it took for starch to be present, that would show me the effect of pH on the activity of enzyme. Is the starch being produced faster if I use a higher pH value, or is it slowing down at a lower pH value? That is what I'm gonna need to find out. That's what I mean by what is the effect of pH on the activity of enzyme. Figure 7 shows an image of an animal cell taken using a microscope with a 10 times eyepiece lens and a 40 times objective lens. Okay, that is the image. That's the nucleus. Question 1. The total magnification of the animal cell is, for 1 mark. So what I know that 10 times eyepiece, 40 times the objective lens. So the total would be 400 times. So my answer would be option C. The diameter of the cell is 15 micrometers. Use figure 7 to estimate the diameter of the cell nucleus. The diameter of the cell is 15 micrometers. So it says use figure 7 to estimate. For 1 mark, so I need to estimate. Okay, estimating. So I go back to the diagram, bring my ruler, measure my actual cell. The actual image size of the cell is 3 centimeters. Okay, the length, the size of the nucleus is 1 centimeter. Okay, so the ratio in this case is 3:1. Okay, so this is me applying my math skills in ratio. So I go back to the question again. To estimate the diameter of the cell nucleus, if this is 3:1 and the cell diameter is 15, my nucleus diameter would be 5. So I have to write my answer, 5 micrometers. So I have estimated. Give the measurement of 15 micrometers in millimeters. So if I bring back what I know, micrometers to millimeters, I need to divide by 1000. That's what I know. So what I do now is divide 15 micrometers by 1000. That gives me 0.015 millimeters. That's for 1 mark. State what is produced when protein is digested. This means broken down. Digestion means to break down. So when protein breaks down, it makes amino acid. And the enzyme that is used is protease. Alcohol is broken down by the liver cells. Broken down by the liver cells for 1 mark. Respiration, no. Osmosis, no. Transpiration, no. So that will be diffusion. If you have found this video useful, please like and leave a comment. Subscribe and share with your friends. The aim of the video is to help you. Until next time.